umbilical cord from the umbilical cord lining, this, and Borden's jelly, which is this part on the inside. So we want to grow new cells because they've got the
important characteristics,
which are that they're pluripotent, so they can grow into any kind of cell. They can differentiate into any cell. They are non-tumorigenic, unlike embryonic stem cells, and they are stress-enduring, so they can grow in like stressful environments. So the way we grow the cells is, first they're obtained from umbilical cords, which are donated by the company StemSite, and we grow them in just in petri dishes with cell medium, which contains like antibiotics and nutrients, which just helps them grow. And so once we grow the cells, to find out if we actually grew the right kind of cells, we use two different methods. We use antibody staining. So this is called immunohistochemistry.
So basically, we take these antibodies that stain for specific markers on the cell, and we just add them to our culture, and then if it fluoresces, then we know we got the right marker.
So we just count them just by looking through a microscope and just counting the percentage of cells that show up green. The other way is by using flow cytometry. We use this really cool machine called the FACS machine, the fluorescence activated cell sorter. So what that does is it basically pushes a stream of cells suspended in fluid past a laser detector, and by measuring the light scattering and the fluorescence of the cells, it separates them into the different kinds. So we had some problems with our antibody staining, so our results have mainly come from this method. And this is what is shown in these graphs. So we're trying to find the cells which show SSCA3, the specific stage-specific embryonic antigen 3. It's a protein which is on the surface of these muse cells. So on this side, these are the ones that showed positive for SSCA3, and we got 30%. Our goal is to get up to 90%.
So basically trying to get these cells in large quantities has been our biggest problem and the biggest problem with this muse cell industry. And that's why we are looking for a new method. And that method kind of comes from the enzyme aldehyde dehydrogenase. Do you want to tell them about that? Okay. So a little bit about this enzyme. It's in a lot of your body's cells, and in stem cell therapy, like a lot of cancer research has shown that stem cells with this specific enzyme can actually withstand adverse cellular environments. So if you transplant something into an injury site, it will almost immediately die off, because cells, they don't like being in like a very like changing environment. When they're being cultured, they're getting all the nutrients they want, they're very warm, things like that. So trying to find cells that are very stress enduring is hard. And Dr. Young kind of theorized that like maybe since aldehyde dehydrogenase is something that's found in cells that are stress enduring, and muse cells are known to be this stress enduring, maybe this correlates. And you want to talk about our results?
Yeah, so 40%, approximately, actually it was 39%, right, were positive for SSCA3, and we found about 30% positive for aldehyde dehydrogenase. So our next step is to find out if those are actually the same cells which are expressing both of those things. And if they are, then we can use a chemical called cyclophosphamine, which will basically kill all the cells which do not have the aldehyde dehydrogenase, and that is a way to completely isolate the cells that we're trying to find.
Any questions?
Yes.
It kind of flies over my head too. Like Dr. Young will be telling me about it, and I'll be like, okay.
So you haven't implanted the cells yet?
So far we have done one transplant. This was last summer, but this was with bone marrow cells. But we found out that bone marrow cells are not the best source of these cells, so now we're using umbilical cord. We do know that these cells are non-tumorigenic though. There have been a lot of studies conducted on it, so we know that they don't cause tumors. But Dr. Young doesn't want to transplant them until we have a big quantity of these cells. So we've been trying to get like 80 to 90% positive. That way we then can transplant them and see what they will do. First uninjured, then injured. And maybe clinical trials.